I'd like to welcome all of you to be here to support Mariem Gabelaias' PhD thesis defense. Mariem started here at Ilyas State University as an MS student. Then she became a PhD student, and ever since she's been quite a prolific researcher. She's done her PhD research at Ilyas State University, Georgia, and Ghent University, Belgium. Now let me really very briefly introduce the defense committee. And before doing so, I want Mrs. Bella Japashvili from Ilyas State University to be my co-chair, and I hope there's no objections to that among the committee members. No objection. So I've been asked to be chair of this committee. My name is Alexander Tawashilishvili. I'm from Ilyas State University. Here we have Zura next to me, Zura Urielidze, also from Ilyas State University, as well as the director of the Tbilisi Zoo. Next, Alexei Yanchukov from Zonguldak Bülent Hecevit University, Turkey. Bela Japashvili, my co-chair from Ilyas State University, and on my right, Cort Anderson, Ilyas State University. And the Belgian side Jens de Meyer, and I hope I'm not mispronouncing it, watching or observing from the screen through Skype connection. Now, a little bit about the format of this defense. First of all, the candidate, Mariem Gabelaia, will give a presentation about her research achievements over the last several years, trying to do it within 30 minutes. And at the end of this presentation, we'll hear comments from two referees. The one is Natalia Kopaliani from Ilyas State University, but she's not present here, and she has provided her comments in a written form, and I guess Mariem will present it to us. And the other referee is from Belgium, Anthony Harrell. Again, am I pronouncing correctly? Hopefully. And he also will hopefully provide a presentation. We have to read. My co-chair has the copy of his, hard copy of his comments. So after that, then the floor will be opened up to questions and discussions. And after that, I'll ask the audience to leave the room, except for the committee members, so that the committee members can discuss any actions that might be needed to improve the work, like missing points, the concepts, and etc. And then the conclusion will be provided to the candidate. With all that said, I'd like to wish our candidate a good luck. So Mariem, you have the floor, and now we can go ahead and make your presentation.
Okay, hello everyone. First of all, thank you for attending my thesis defense. And today I will tell you briefly about my study, which was done in the framework of my doctorate studies, phylogeny and morphological variation in the rock lizards of the genus Darevskia, which is mainly about linking the genetical and morphological variations of this species group. Before I start with the topic, I would like to briefly introduce the species itself. They belong to family Lacertida. They are quite diverse. They have up to, more than 30 species, from which 26 are bisexual and 7 parthenogenetic forms. Also, even though most of them are rock dwellers, they have 5 ground-dwelling species. They are quite small. Their body length ranges from 55 up to 85 millimeters, and morphologically they are quite similar. They have multiple overlapping scalation characteristics, thus they are very difficult to distinguish from each other. Taxonomy of Darevskia was first studied by Darevskia, where he used multiple body measurements of the lizards and scalation traits, and revealed only 6 species based on his study: Praticola, Derugini, Saxicola, Caucasica, Mixta, and Rudis. As for other currently recognized species, they were considered as subspecies of those 6. Darevskia also based the phylogenetic relationships of these animals based on qualitative morphometrics, and until 2000s their phylogeny was based on Darevsky's study. In 2000, Murphy with his co-authors used a mitochondrial DNA analysis, and he revealed 15 bisexual species belonging to 3 monophyletic matrilineal clades: Rudis clade, Caucasica clade, and Saxicola clade. The phylogeny of Darevskia is largely still based on this study, with one exception of Darevskia parvula, which was placed in Rudis clade in Murphy's study, but later studies, including larger fragments of cytochrome B analysis, placed him rather basally. Thus, based on these results, it was considered that morphological data of Darevskia is useless for reconstructing phylogenetic relationships. Based on that, we consider that using other various morphological methods, we would be able to find the traits of morphological variation, which would correlate with their phylogenetic relationships. Also, we tried to find the morphological components which were species-specific and conservative for delimiting their boundaries. For that purpose, we collected up to 415 individuals. Some of them were collected by the researchers of Institute of Ecology, starting from 2008. Later in 2012, I also joined the research team and collected some more samples throughout Georgia. Overall, we have 13 species belonging to 4 matrilineal clades of Darevskia. As for the methods used for genetic analysis, we used mitochondrial DNA analysis. For morphological ones, we used qualitative traditional morphometrics, which was also used by Darevsky, and 2D and 3D geometric morphometrics. Well, we focused on geometric morphometrics because multiple studies have shown that it is more powerful in assessing shape variation rather than traditional methods. So to briefly explain how the methodology was done, for mtDNA analysis, we collected the tail tips of the lizards. We extracted DNA using the standard protocol from the Kiagen manufacturer. After PCR reaction and sequencing, we edited and aligned the sequences and prepared them for the further analysis. For traditional morphometric analysis, we studied up to 20 scalation characters and measurements. The values included the number, the presence or absence, and the configuration of the morphological traits. For 2D geometric morphometrics, we used elliptic Fourier outline analysis in software shape. So how it is done, we first trace the outlines of the study object. Then after importing them to the software, they are transformed in chain codes. Then after normalizing size and orientation of the outlines, they are transformed to elliptic Fourier descriptors, which are the descriptors of the shape. And later on, principal component analysis is done on these elliptic Fourier descriptors in order to find the shape dimensions which significantly vary among the individuals. For 3D geometric morphometrics, we collected live individuals. They were anesthetized using chloroform and placed in the tube where their head was pointing up. Then we photographed them 36 times from two perspectives, 90-degree perspective and 45-degree perspective. And the camera by each shot was moving by 10 degrees. The resulting 2D images were then imported to software Agisoft, where we constructed the 3D model. And I will briefly show you how the models look. Shortly, like this. Later, we digitized three-dimensional landmarks on the homologous scalation character intersections. And later we applied principal component analysis for extracting the variable shape components. So we will start first with phylogeny versus morphology. As long as morphological data of Darevskia was considered useless for phylogenetic purposes, we tried to apply geometric morphometrics and see if some shape components of the scales were correlated with phylogeny. For that purpose, we chose 6 species belonging to 3 metrilineal clades of Darevskia and applied elliptic Fourier analysis on the pileu shape and anal scale shape. As a result, out of pileu shape and anal scale shape, only anal scale shape confirmed significant phylogenetic signal. We quantified phylogenetic signal using a Blomberg scale, which is a measure of statistical dependence of animal's morphological trait with their phylogeny. It was resulted as 1.5, which means that the similarity among the traits among different species are more similar than it is assumed under Brownian evolution model. This same phylogenetically informative trait significantly differentiated the main major clades of Darevskia, but could not separate the individual species. And this variation was associated with high and convex versus short and concave anal scale. Thus, overall, we can say that it was geometric morphometric was able to better assess the phylogenetic relationships through morphological variation than traditional morphometrics. In addition, we also used the median scores of each principal component per species and constructed the UPGMA tree, which resulted in a topology which is congruent to the mitochondrial tree of the species. For our next study, even though pileu shape did not show any phylogenetic relatedness, we tried to quantify it using 3-dimensional morphometrics and see if the inclusion of all 3 dimensions of the shape, it could provide components correlated with phylogeny. We also used 2-dimensional shapes of anal scale, 5th upper labial scale, and head frontal scale. And in addition, checking for possible convergences in the shape data. We tested 3 different convergent factors for our shape data: habitat type, size, and sex. For habitat type, it showed that the largest variation in the head shape was associated with head height, high versus short flat head. Size was associated mostly with the breadth of the lower jaw and anal scale height, and sex was associated not with phylogeny as we saw in the previous study, but with sex and a concave convex anal scale shape. But all the traits were phylogenetically constrained, thus did not produce convergent patterns except the habitat type. We can easily see this on the picture. For example, we tested 3 different groups, the strict ground dwellers, strict rock dwellers, and the species with intermediate life mode. The differences in shape is quite obvious even from the pictures. The ground-dwelling Daryugini, which is close relative of Darevskia mixta, which is a rock dweller, were more different than morphologically than the Darevskia mixta and Darevskia porchinsky, who are distantly related. For assessing phylogenetic signal, in this study we used Blomberg's K as well as Pagel's lambda, which is another measure of phylogenetic signal. And out of all the components, only 3D head shape, a 4th principal component, showed the association with phylogeny for both of these coefficients. And the phylogenetic relatedness was associated with the flatness of the rostral area, with the width of the jaw, and the relative length of the interparietal scale. Even more, after we excluded the habitat, size, and sex-related components from the shape data, we were able to construct the neighbor joining tree using discriminant analysis first and identifying the discriminant factors, which was quite similar to the tree based on mitochondrial DNA. Thus, we can say that geometric morphometrics was able to find the shape components which were associated with phylogeny, which traditional morphometrics or 2-dimensional geometric morphometrics was not able to do. The last 3 chapters of my thesis are associated with finding links among genetic and morphologically defined species boundaries. For our first study, we studied Darevskia mixta's phylogeographic pattern and the morphological variation. We collected the samples of endemic Darevskia mixta throughout its distribution, from Greater Caucasus and from the Lesser Caucasus. We quantified, did the analysis of mitochondrial DNA and studied its scalation characteristics, overall 18 scalation characters. As a result of genetic analysis, the maximum likelihood tree resulted in 3 monophyletic matrilineal lineages, 1 from the Greater Caucasus and 2 from the Lesser Caucasus. Both genetic data as well as morphological data showed that the origin of Darevskia mixta happened on the Lesser Caucasus because morphological variation was higher in Lesser Caucasus, as well as genetic data variation. Even though 4 morphological characters were significantly different between these 2 populations, the analysis was still not able to identify the individuals, and there were still multiple overlaps among the individuals of Greater Caucasus and Lesser Caucasus. For our another study, we tried to assess Darevskia brownery's taxonomical status by studying the populations of Darevskia brownery in Georgia, which have not been studied before when assessing their phylogenetic or morphological variation. We collected the samples from Enguri Valley and from Tehuri Valley, and used mtDNA analysis to assess their genetics. Also, we used traditional morphometrics based on 4 scalation characteristics, which was also used by Darevsky when he studied the morphological variation of nominal Darevskia brownery. As a result of genetic analysis, it resulted in 3 equidistant mitochondrial clades of Saxicola, within Saxicola clade. The one was Tehuri Valley lizards, which were referred as Darevskia brownery before, but are indeed more distant from nominal Darevskia brownery than from other species such as Darevskia saxicola or Darevskia lindholmi. The another clade was consisted of Anguri Valley lizards and Darevskia sherbaki, another subspecies of nominal Darevskia browneri, which has been seen to be separate species from nominal Darevskia browneri from recent genetic studies. And also nominal Darevskia browneri and Darevskia lindholmi. So here are two possible scenarios in this case. Either we can say that all these lineages are the polytypic version of one species, Darevskia saxicola, and this species can be referred to subspecies of Darevskia saxicola, or we can accept that if nominal Darevskia browneri, Darevskia lindholmi, and Darevskia saxicola are different species, then the Georgian populations from Tehuri and Anguri Valley are the equidistant from Darevskia lindholmi than from Darevskia browneri. Thus, Georgian populations cannot be referred as part of Darevskia browneri nominal species. The morphological variation was also not in line with their phylogenetic pattern. Tehuri lizards did indeed show that they had slightly broader heads than the rest of the clade saxicola, but Anguri population showed to be morphological similar to earlier described Darevskia browneri rather than to Darevskia saxicola, which it was closer in phylogenetic tree. Thus, once again, traditional morphometrics was not able to fully follow the species phylogenetic pattern. In the end, as long as two-dimensional geometric morphometrics and traditional morphometrics were not able to fully differentiate the closely related species, we tried to see if 3D geometric morphometrics would provide more shape dimensions which could separate these close related species. For that purpose, we chose Rudis clade, which consists of quite closely related species. Their lineages diverged somewhere in late Pleistocene, and they are still in the stage of lineage sorting. In this study, Darevskia rudis was represented by two subspecies, Darevskia rudis obscura and Darevskia rudis rudis, and then another species, Darevskia valentini and Darevskia porchinsky. Their distribution is as follows. Darevskia obscura has the intermediate distribution among all the rest, has contact areas with the rest of the Rudis clade species, and quite often hybridizes with them. After applying principal component analysis to 3D shape data, it showed that Rudis obscura individuals and Rudis rudis individuals, which were considered as one species, are completely different morphologically based on their 3D head shape, even more than Darevskia rudis obscura from Porchinsky or Valentini. And this is also in line with the earlier genetic studies which showed that Darevskia rudis obscura has a monophyletic origin, matrilineally. In order to see if 3D GM would be able to significantly separate all the studied species in Rudis clade, we applied between group principal component analysis, which could separate the species with minor overlaps. Thus, we can say that 3D geometric morphometrics was more powerful in separating closely related species and delimiting their boundaries than 2D geometric morphometrics or traditional morphometrics. Also, Rudis obscura was equidistant from the Darevskia rudis populations as from the rest of Rudis clade species, and because of that reason, we suggested to elevate the status of Rudis obscura to species level Darevskia obscura. As a summary, based on my study, we can say that it is possible to extract phylogenetic signal from Darevskia shape data using geometric morphometrics. And it is even possible to reconstruct the species phylogeny, the tree which is closely similar to species phylogeny, once the convergent patterns are removed from the shape data. Overall, geometric morphometrics outperform traditional morphometrics, but 3D GM was the most powerful for assessing the species phylogeny and for separating the closely related species. For the future work, we hypothesize that the skull shape is less prone to homoplasy and is more conservative and species-specific for delimiting the species boundaries. Thus, we want to try to use landmark-based as well as landmark-free analysis and compare it to the results of the external head shape analysis, which we have used in the frame of my doctoral study. And yes, in the end, I want to thank my supervisors, first of all. Also, everyone who has contributed to my research, Mari Murtkhaldze, Tamar Beridze, Gocha Golubiani, Anna Valiani for genetic help in the genetic work. Also, Natia Georgi, Levan Muladze, Levan Inua, David Temur, and also Alexandre for helping with collecting the samples. Also, Wolfgang Bome and Kurt Anderson for their suggestions and reviews of the papers, and my friends and family and everyone who has supported me. Also the reviewers for their helpful comments. University administration, of course, who made this joint study possible and followed me in every stage. Institutions and, of course, funding agencies which financed my studies. So, thank you.
Thanks. Excellent. You did it within like 25 minutes. You have 5 more minutes if you want to say something else.
Thanks.
Okay. Now let's hear the comments from the referees. So I probably asked Mariam to
Yes.
Comments from Natia Valiani.
Yes, so it was in Georgian, but I will try to translate it as well as possible. Yeah. So, according to her review, the study subject is not the newest, but it provides the new insight into the morphological and genetic variation within Darevskia species. Also, the study goal was quite obvious from the thesis. The literature has been extensively reviewed, and geometric morphometric methods, which are newer methods of studying morphology, were worth mentioning. She had a couple of questions also. Should I read them and answer? Or maybe...
well, I don't think it's necessary. We all want to hear, do you address those concerns that Natia has?
Well, these were the questions rather than suggestions to change something, so I can answer them. Like she meant how correct it is to extract the components, shape components which are related with convergent factors rather than phylogeny. So I can only answer that we did it for a sole reason, to see if we were able to reconstruct a tree which would be similar to phylogenetic relationships. Otherwise, of course, we considered the convergent shape components in other analysis done. Yeah. And there are a couple more questions if...
Well, then there are now comments for urging any changes or alterations, so...
No, I don't think
we are
so.
good. So let's now, I'm going to ask my co-chair to...
Should I read or
if you find any...
I got it's really what the defense, but I try to summarize. In general, the referee gives the highest score, summa cum laude, excellent, and in his review he emphasized and that and showed that Mariam is capable to do excellent scientific work and produce high quality publication, which she already done while she published already chapter 4. Yes, am I right? Yeah, for chapter 4.
This one was not published.
Yeah. And also chapter 3 has been submitted for publication. Also he is giving an excellent mark. He found some weak points, maybe let's say. So it's related writing in English, and also he has several questions. I will read this part, okay? I was surprised you did not test for difference in diet between species. It is known that this is also an important driver of feed shape of their dietary label. And also he's pointing about this question related to your methods of statistics, why ideally you want to run a maneuver instead of ANOVA, and also the final remark is related to the algorithm that you use, the ICP algorithm to perform the 3D geomorphometric on head shape. And I'll read this part. I was a bit surprised by this suggestion, as using this algorithm results in loss of biological homology in the structure compared. So my question is, how useful is this approach really if you want to extract biologically relevant features like phylogenetic structure, etc. And finally, he says that it's just suggestions or some remarks, and he is given excellent summa cum laude.
I just, you have to go over this again, you know.
Yes, maybe I will start from the last one. Yes. Okay, I will start with the first one. As for the diet, there have not been much solid studies about the dietary preferences of Darevskia lizards. There are some works done, but mostly they show that these lizards eat everything, and it largely depends on the prey size rather than any other preferences. So the bigger lizards, which are, for example, Rudis or Valentini, eat maybe bigger prey, but also smaller ones. So the size of the prey is the main factor for dietary differences in this species, most probably.
Sounds like we have basically we have a clearance from referees. And before I ask the audience to leave, for us to make a final decision, to discuss a final decision, is there anybody in the audience who wants to say anything in Mariam's support or to oppose to whatever she presented?
I just want to say thank you, Mariam. It was excellent work, and it was very nice presentation. So congratulations. This is the only remark what I have to say.
Thank you. Should I answer the other questions from the reviewer? Or?
I think there's no need.
Okay.
Unless you haven't covered them in your presentation already.
No.
In your detailed presentation.
No, I have not.
You haven't covered.
It's just the ICP algorithm is for future plans. So we have not used it yet, but we want to compare it with the landmark-based approach and see if it can provide some additional shape components which are not as accessible using landmarks. Yeah.
I was just going to say, we might like to ask our colleagues again if they have any questions or any commentary at this point.
Yeah, Mr. Ford is suggesting we should ask our colleagues from Belgium to comment on the presentation before I dismiss the audience.
Pfizer, I don't know. things I think with Mariam, I can only all the work that especially one proved to be proficient at getting into new little inputs and can only but Jens has some comments here.
Should we recall?
I really enjoyed it. It's a very nice piece of work.
Maybe we can recall because there's a problem with connection.
Can you hear us?
Now, yes?
Now we can.
Can I speak? Or
Yeah.
Try please. Go ahead.
So Mariam, I enjoyed reading your. It's a very nice. It has.
Oh, again, the connection is
Maybe we can call again.
funny.
I will try to call once again. Maybe it will.
Disable the video, maybe. Okay.
Okay. I will disable the video for a while.
Yeah. Okay. Yeah. I'll do the same here.
Okay.
Is it better?
I think, yeah.
Yes. Please try again. Sorry for this technical glitch.
Okay. So for the third time, Mariam, I really enjoyed reading your PhD. It's a very nice piece of work. I don't have any big comments, but just I am a functional morphologist. So there's a point where you're looking at the effect of you want to look whether there are differences in morphology related to ecology. And I was wondering what difference you would expect in scaling depending on the ecology of the lizards.
Well, I would expect the one like the difference in their habitat types, the rocky, for example, some lizards which are ground dwellers, they live in the meadows or valleys, and they do not need rocky habitats for egg laying or for their just for most of the time. As for the rock dwellers, they have flatter heads because they need to hide in the narrow rock cracks, and they also lay eggs inside these cracks. Thus, the flatness of the body and of the head is the most was shown by the study to be the biggest variation, and I was expecting that as well. Do you mean other than that, or in general?
No, that's fine. Thank you.
Okay, thank you.
Now I guess we have to discuss and make a final decision, to have a final verdict on Mariam's performance. I think I would, as a co-chair, I would go first, and I would give the top grading.
It's an excellent presentation. A lot of information, a lot of work. No, it's absolutely a good, highly qualified dissertation.
Yes, we all agree. So let me see this Latin thing. Summa. There's also. Summa and magna cum laude. And there's also cum laude. So, what do the Belgians think?
Am I supposed to advise as well? Or
Yes.
Please, could you repeat it? Could you repeat it?
Am I
Well, again, we are having some.
feedback?
Yes, please, if you.
So, correctly, you're just give
Summa cum laude. Summa cum laude. Yeah, okay, so then summa cum laude, we all agree on that. So that's
It's a really good piece of work. Well, we're not working with grades in Belgium anymore, so it's a bit difficult to make a distinction between summa cum laude and
Same here, don't worry. I think the summa is the top. We all agree on that, right? Summa is the top.
Summa is absolutely better than magna.
Better than magna, yeah.
summa then.
Summa then, yes. We all want this top one, so I'll check in with the specialists here too. So we all give the grade top marking, right? Grading. Okay. So that's about it then. I'm gonna have the audience back, and we're gonna announce this final decision. So thanks a lot. If you wanna say
No, we're good.
anything.
My question is for our chair people. So are we not going to grill the candidate at all? We're not going to ask her questions about her work? Okay.
There's no necessity for that.
We had to ask also another supervisor, to say something. So
When they come back in, they will. Yeah, when they come back, yeah. That's how it goes. Okay, then we're going to have the audience back. So welcome back. So to cut to the chase, we all agree, we all converge on one decision: the top grading, which is summa cum laude. And now, you guys did not question her at all, but you have to say a few words. Students, I strongly urge one or two of you to say something about her, about Mariam. Please. No? Students, I want her students and her friends. Please, go ahead.
She's a great example for me, and she's always very helpful when we other PhD students need some suggestions or help. She's very helpful, and also, yeah, she's a great friend. Besides the science and help and some science talks or anything, and yeah, this is, I think, all I can say.
Excellent job, Mark. Thank you.
Okay.
Now I guess Mariam's Georgian supervisor wants to say a few words.
I will be very brief because, well, Mariam appeared at our university, I think, in 2012. It was there were some wonders because actually students which start masters in ecology supposed to have biological education, but I think that it was a very proper decision to take her on the faculty after her bachelor in physics, because it's somehow, maybe I'm wrong, but it is somehow associated with a very clear way of thinking. And well, it was not mistake because Mariam showed herself a extremely desperate person. So a person which can be asked to do a research, asked to do a work, and you would not worry about this, you would never worry about this, so you may be sure that everything will be done in the best way. And a person which very easily understands complicated stuff and actually spends some time until she does it, but you know, it's like doing this like a very simple work, which is not very useful. I am proud that I was supervising her PhD, and I am proud that actually it was a PhD on a subject which is also very interesting for me. So thank you.
Now I guess, Mr. Dominic, would you like to say a few words? Maybe you should disable the connection, video connection. No, court is typing right now. They can text their final, final few words if they want.
Sorry, the connection was lost.
Hello.
Hi, Dominic. Hi. Can you hear us?
Should I turn on the camera or?
Could you read us? I knew this was coming. Mariam, can you touch that? We just want to hear a final few words from that, and that's it.
Yes, we can hear you.
Could you read us?
Yes, we can hear you. You can
Ja, waarschijnlijk dat dit regelt is al. Ja. Okay, so Mariam, apparently you can hear us, but unfortunately I cannot hear you, but that's not important at this point. I think I can only wrap up this very nice defense with just saying that you did a very nice job, a great job. It was a pleasure having you over in the lab and collaborating with you on a topic of which I actually knew nothing until now. I know a little bit about Darevskia lizards and things related to that. But as I mentioned earlier, I was very much pleased with your working rigorousness and your efficiency at which you very efficiently absorb like a sponge information, very little information actually, to get worked into the other methodology being geometric morphometrics, which was new to you, and you immediately translated that in data that was very useful to very quickly, very efficiently get this worked out in a manuscript that was published, and then even another one, and then one in submission even now. So I can only completely and utterly congratulate you on the work you have achieved, well deserved, and also with a very nice grade. I hope we can stay in contact and collaborate in the near future to continue to do so, and I really will with your scientific or other professional career, and I hope we can meet again soon in whatever condition. See you later.
Thanks. Anyway, you all heard that, and myself, I'm going to also say just a few words. She's been one of my the most brilliant, excellent students, and not to make others jealous, but she's really something. The first ever the class that I had with her, I knew she was going to be an academician, scientist, if she wanted to. So she had all the brain, all this analytical capabilities. That's right now. That's what we are witnessing right now. The proof of that. Thank you, Mariam, and I want to wish you every success in your life.
Thank you so much.
Thanks.
Thank you, Mariam.
And thank you all for coming to support her.